Good morning, dear students. A warm welcome to all of you for our today's online session for the subject Science and Technology Section B. Yesterday, we have completed with the revision of all the 4 chapters for both the sections, and we have also started with a new chapter from Section B, Useful and Harmful Microbes. We will first revise the topic which we have studied yesterday, and then we will move towards the new topic. Yesterday we had seen what we mean by microbes and what are useful microorganisms. Useful, when you say, then the microorganisms or the microbes around us which are useful to mankind, they are termed as useful microorganisms. The two useful microorganisms which we got introduced to yesterday was lactobacilli and rhizobium. Lactobacilli, look at the word lacto. Lactobacilli are the bacteria which are minute and rectangular in shape, which can be seen only under the microscope. Now, they are also called as anaerobic bacteria because they produce energy without the use of oxygen, in the absence of oxygen. The word lactobacilli because they prove very important in the dairy industry as they convert the lactose, the sugar which is present in milk, into lactic acid by the process of fermentation, and hence they are extremely useful in the manufacture of milk products like yogurt, buttermilk, ghee, cheese, also for the large scale production of cider, cocoa, pickles of vegetables, and n number of uses. And due to a variety of uses for human beings, they are called as useful microbes. The next useful microbe which we had studied yesterday was rhizobium, which is also known as symbiotic bacteria. What is rhizobium? Rhizobium is a bacteria which is found in soil that helps in fixing nitrogen in leguminous plants. They are rod shaped as they belong to the Bacillus group. We had seen yesterday rhizobia, which lives in the root nodules. They supply nitrates, nitrites and amino acids to that plant, and in exchange what do? They get the energy in the form of carbohydrates from it, and such a mutually beneficial relationship is called symbiosis, and hence the name symbiotic bacteria. This rhizobia, they produce the nitrogenous compounds from your atmospheric nitrogen. But for this process of nitrogen fixation, they require leguminous plants, and the beans, pulses, they are rich in proteins due to the nitrogenous compounds which are made available to them by rhizobia. After harvesting a leguminous crop, the leftover roots and the other plant parts are deliberately dumped in the farm soil to maintain its bacterial population. Also, we had seen that this use of rhizobium has helped to reduce the use of chemical fertilizers and thereby lower their adverse effects. And really, children, it has helped the farmers in the agricultural field to reduce the expenses on fertilizers, and really it works as a big benefit for the farmers. Also, we had seen one topic, rhizobial inoculation, where nowadays seeds are coated with the rhizobial solution or powder before sowing them. What happens due to this? After sowing, rhizobia enter the plantlets, which is known as rhizobial inoculation. And this experiment, it has helped in the supply of nitrogen to cereal and other crops besides the leguminous crops. You know, rhizobium, they require the roots of the leguminous plants to supply this nitrates, nitrites, etc. But this rhizobial inoculation, due to it, the other cereals and crops besides the leguminous crops, they also get the supply of nitrogen. Today, our new topic, the new useful microbe is yeast. You might have heard about the word yeast, or you are well introduced with the bread which you eat in your breakfast. What is this yeast? This yeast is a eukaryotic single-celled, that is unicellular, and classified as the member of the fungus kingdom. Means it is a fungi. It is a unicellular fungus with 1500 different species in existence in this universe. It is a heterotrophic fungal microbe that grows on carbon compounds. And what happens here? Yeast, it grows and multiplies very quickly due to the carbon compounds in the sugar solution. Reproduction mode is asexual by the process of budding. Budding means you have seen potatoes. If you keep in the house for large number of days unattended, after some period small yellowish colored buds appear on its top. Similarly here, in the case of yeast, small round bodies attached to the parent cells, they are known as the buds. These are the new daughter cells of the yeast in the process of formation. So reproduction is by the process of budding. As I have told you, it grows and multiplies very quickly due to the carbon compounds in the sugar solution. And in the process of obtaining its nutrition, yeast cells, they convert the carbohydrates in that solution into alcohol and carbon dioxide, which is termed as fermentation. Now this yeast is used to cause fermentation and leavening, and in the making of bread, alcoholic drinks, biofuels. So yeast has n number of uses. Even brewing wine and beer fermentation, here the mixture is also used. In this, the yeast is used for centuries to make it alcoholic. So yeast, it also works by serving as one of the leavening agents in the process of fermentation, which is essential in the making of bread. You can see here, to make the bread soft and fluffy. Now this all you will not get in the textbook. This all pictures I have taken from the internet. So the main function you can see here is it works by serving as one of the leavening agents in the process of fermentation, which is essential in the making of bread. The purpose of any leavener is to produce the gas that makes the bread rise. Otherwise the bread will not rise, it will not be soft and fluffy. Yeast, it does this by feeding on the sugars in the flour and expelling carbon dioxide gas in the process, due to which the breads, you can see them rising. Many yeasts are sac fungi, including the common yeast used to make the bread. Yeast uses sugar as the food to produce carbon dioxide and alcohol. Trapped carbon dioxide bubbles causes dough to rise and the bread becomes very soft. So the main use of yeast is in the bread making process. Bioremediation is a very small topic which you just have to go through on page number 91. A yeast, Yerrovia lipolytica, is used to absorb the toxins released during the production of palm oil and the heavy metals and minerals released in some other industrial processes. Saccharomyces cerevisiae is used for the absorption of a pollutant, arsenic. Oil spills in the oceans are cleaned with the help of alkanorex bacteria. Our next very important topic for you is antibiotics. Antibiotics. What do you mean by this term? Anti means against, biotics means against life. The one that kills the germs in your body. It is a drug. Any drug that kills the germs in your body is termed as antibiotics. In science, how you can define this antibiotics? the carbon compounds which are obtained from some bacteria and fungi for destroying or preventing the growth of harmful microorganisms are called antibiotics. You know how important antibiotics, how crucial role they play when you are suffering from various diseases, including your small, or you can say the primary levels of cold, cough, up to the high level diseases like typhoid, malaria, etc. And in all these diseases, the main thing which is recommended by the doctors is the use of antibiotics. And really, the introduction of this antibiotics, it has brought about a revolution in the field of medicine. What is this antibiotic? It is a type of antimicrobial substance active against the bacteria. It is most important type of antibacterial agent for fighting the bacterial infections, and antibiotic medications are really widely used in the treatment and prevention of many types of infections. Now, antibiotics, if you see, they mainly act against bacteria. Some are the antibiotics which destroy the protozoa. Now, these antibiotics are classified into two types. One is the broad spectrum antibiotics. Broad spectrum antibiotics. What do you mean by it? Some antibiotics, they are useful against a wide variety of bacteria. These antibiotics, they play a major role against variety of bacteria, and hence broad, wide number. Hence they are called as broad spectrum antibiotics. And the different examples of broad spectrum antibiotics are ampicillin, amoxicillin, tetracycline, etc. If you search for these names on the Google net, you will be able to see. Amoxicillin is, you are familiar with the term mox, tablets which are given by the doctor when you suffer from high fever or cold and cough. The next is narrow spectrum antibiotics. Now, when the pathogen cannot be identified, even though the symptoms of the disease are visible, then broad spectrum antibiotics are used. Means you cannot identify the pathogen, but the symptoms of the disease you can see. Then you can make use of broad spectrum antibiotics. And narrow spectrum when you can make use, when a pathogenic microorganism is definitely known that this microorganism has caused this disease, then you can make use of narrow spectrum antibiotics. And the different examples of narrow spectrum antibiotics are penicillin, gentamicin, erythromycin, etc. And the use of this antibiotics, it stops the infection which are caused by different bacteria. It kills or inhibits the growth of bacteria, and thus they are called as life-saving drugs, and has really brought about a great, you can say, relaxation, a big revolution in the field of medicine. The next topic is penicillin. Penicillin is nothing but, again, it is a group of antibiotics which are obtained from a fungus, Penicillium. Penicillium. It is used for controlling the different bacterial infections which are caused by different bacteria like Staphylococci, Clostridia, Streptococci, etc. Now, medicines which contain penicillin, they are useful to treat certain bacterial infections of your ear, nose, throat, skin, as well as also diseases like pneumonia and scarlet fever. It is termed as a narrow spectrum antibiotic. Now, who introduced this penicillin? Before moving ahead, as I have told you, use of antibiotics, it has brought about a big change, a big relaxation, a revolution in the field of medicine. But there are some cautions which you have to take while consuming this antibiotics. That cautions which are given in the textbook, we will just go through it. You don't have to study or something, but you should know, as it is a very important fact. Antibiotics should be taken only when prescribed by a doctor. Even though you know that XYZ, it is an antibiotic which is used to cure the fever, but still, unless and until a doctor, a physician, it allots that tablet to you, you should not consume it on your own. Yes, there are certain antibiotics like crocin or Vicks Action 500, which they are very mild in nature, and in the initial stage, suppose you are suffering from high fever during the night time, to lower the temperature, initial level you can consume 1 or 2 tablets till you visit the doctor, but you should not go on consuming it on your own. Don't purchase any antibiotic from medical stores without a prescription from a doctor. Unless and until the doctor gives you a prescription, do not buy anything and consume on your own. Don't consume antibiotics on your own to treat common diseases like a throat infection, common cold or influenza. Many people, they avoid going to the doctor because they know after going to the doctor, then heavy dosages of medicines are given. They go to the chemist, they buy antibiotics on their own and consume them. It is really dangerous because you are not the doctor. You do not know what actual disease you are suffering from. So initially you might get rest, but somewhere the bacteria or the infection can retain inside your body and it can prove fatal ahead. So visit the doctor, get diagnosed, and then only take the medicines. Even if you feel well before completing of the prescribed course of the antibiotic, you must continue and complete it. When you go to the doctor, a course of medicines is given. Per day, 3 dosages are given. Multiply by 2 days or 3 days depending upon the severity which you are facing. What you do? After consuming 1 or 2 doses, you feel relaxed, you feel good, you feel better, and then you avoid continuing the, you can say, complete treatment. Don't do that, because that particular treatment is given by the doctor, because the doctor knows how much amount of infection you are having. Might be inside your body. So we will take an example. 100 viruses have attacked. You have taken 2 dosages. Doctor has given 3 dosage per day, multiply by 2 days, 6 dosages. Out of 100 virus, after taking 2 dosages, 50 have died, 50 are killed. So you might start feeling better and you will discontinue the course. What happens? The remaining 50 which are there in your body, they can attack your body further and then it will become very dangerous. So whatever course is allotted by the doctor, in that particular course, your entire infection goes away. If it doesn't go away, then the doctor will diagnose for further different types of infections and can give you high dosages of medicines. So do not discontinue the course. Don't suggest to others the antibiotics which were useful to you. Many a times you think you all are doctors, and then whatever medicines you consume and are good for you, you suggest others, which may, some of the people, they may be allergic to different antibiotics. So without the recommendation of the doctor, you should not suggest any antibiotic to other people. Now let us come towards penicillin. You can see here a picture of Dr. Alexander Fleming. We are in the topic of penicillin. Here now this Dr. Alexander Fleming, he was a professor of microbiology at St. Mary's Hospital and had cultured varieties of bacteria and fungi in petri dishes in his laboratory. He really, on September 3rd, 1928, while observing staphylococci cultures, made an interesting observation in one of the petri dish. In that petri dish, fungal colonies had grown, but the area around these colonies was very clear. The area around these colonies, it was really very clean and clear. That is what had happened over here. The bacteria had actually been destroyed. After further studies, he confirmed that the fungus which was growing there was Penicillium, and its secretion had destroyed the bacterial colonies, and thus the first antibiotic, that is penicillin, had been discovered accidentally, and his, you can say, discovery, this formed the basis for the attempts to find cures for incurable diseases. And really for this, whatever you can say accidental discovery or the, you can say, advantage which is given to us by Dr. Alexander Fleming in the form of penicillin, we should be really grateful and thankful to him for discovering such a very important life-saving antibiotic. With this, we complete with the first half of the chapter, that is useful microorganisms. Now we move towards the next part. As we say, every coin has two sides. Harmful microorganisms. Harmful. The word itself tells you that the microorganisms which cause diseases, which harm not only the human beings, plants, animals, they are termed as harmful microorganisms. The first is fungi. Most of the fungi, if you see, they are saprophytic and not pathogenic to plants, animals and human beings. Most fungi, they are pathogenic to plants. They are saprophytic, they are not pathogenic. However, a relative, you can say few fungal species, they are phytopathogenic and they can cause diseases in human and produce, you can say, different infections, allergies which can affect the plants, animals and human beings. Now microscopic spores of fungi, they are present in the air around you. If there is sufficient moisture, see, for this fungi to, you can say, reproduce or grow, there should be sufficient amount of moisture. Then the spores will germinate on cotton fabric, gunny bags, leather, wooden items, etc. If you have seen, I don't know how many of you have seen, but this leather articles and all in the rainy season, if left unattended and after a few days, if you just come across them, small fungi, white colored fungi, you can see growing on them. How they grow? The fungal hyphae, they penetrate deep into the materials like leather, fabric, etc. to obtain the nutrition. They get the nutrition from it and to reproduce. This causes the materials to wear and become weak, and as a result, you will see the gunny bags or the leather items like shoes, purses, belts, etc. on which the fungi have grown, they do not last long. Even wooden items, they get spoiled. So one of the harmful microorganisms is the fungi. Even you might have noticed a black powder, you can see here, or white discs floating on the pickle or muramba when a jar is opened after a long time. What exactly is all this? Why are such food items not good to eat? You might have seen on the jams, pickles, if they are not opened for a large amount of time and if any type of moisture is there around them, especially during the rainy season, such black colored particles or white, you can say, discs are seen floating on them. So various species of fungi, they grow on food items like pickles, muramba, jams, sauce, chutneys. They use the nutrients in these food items. These food items, jams and all, they have the moisture present in them. So this fungi, they will use the nutrients in these food items for their growth and reproduction, and during this activity, what they will do, they will release the mycotoxins, which are the poisonous chemicals, into the food, and thus if you find your food attacked by such type of fungi, then it is very dangerous to consume the food because it becomes poisonous. Hence the food on which such fungi have grown, it is not fit for eating. The next harmful microbe is Clostridium. Clostridium are really the harmful bacteria which spoil the cooked food. Sometimes you might have heard about the cases of food poisoning which occur during community feasts. How does food become poisonous all of a sudden? It is because of the attack of this harmful bacteria, which are rod shaped and produce bottle shaped endospores in adverse condition. Out of the 100 different species of this bacterium, some are free living in the soil, Some live in the alimentary canal of humans and other animals. Now, one special characteristic of this bacteria is that they cannot withstand the normal oxygen level of the air because they grow in anaerobic conditions, that is, in the absence of oxygen. So, due to the attack of this Clostridium, the main pain areas is in the abdomen. You have abdominal discomfort which leads to vomiting, diarrhea, bloating, fever and life-threatening inflammation of the colon. Colon means the longest part of the large intestine. So really they are the harmful bacteria, and especially they are the ones which spoil the cooked food and hence attack large number of people and lead to food poisoning during community feasts. You might have heard. Other harmful microorganisms are also there. It is not only the Clostridium which cause illness, but other kinds of microorganisms like the bacteria, viruses, protozoa, fungi, they are also responsible for the different diseases that affect the human beings. You might have learned about the viruses which are smaller than bacteria and can grow and reproduce only in living cells. Now you have one chart in your textbook on page number 94. You just have to go through the chart, the names of the diseases, the pathogen which attacks, the mode of infection and the preventive measures are given. Just go through the chart because last year it had been removed as the reduction in syllabus. So the ones which you feel really important like AIDS or pneumonia, malaria, that you can just go through. With this, I declare that the explanation for chapter number 8 is over. There is still some amount of time, so we will just complete the text. Start with the textual exercise. I request all of you to just keep your textbooks open. At least we will complete question number 1 today. Complete the statements with proper option from those given below and explain the statements. Here you don't have to just fill in the blanks, but whatever answer you are giving, whatever option you are selecting, you have to give the explanation. The options are given in the bracket: mycotoxins, budding and rhizobium. Let us see the first one. Yeast reproduces asexually by, just now we have discussed when we have studied about yeast, it is by the budding method. So this explanation you can mark in the textbook on page number 90. Page number 90, you can see figure 8.3, yeast cells. Near that you have the heading as activity, first, second, third, fourth paragraph. Some of those cells may have. There you can just start. Yeast cell develop small round bodies on the parent cell. These are called the buds, and new daughter cells develop from this buds. You can just modify the points given over there in the textbook and you can write the explanation. Now whatever explanation here I am giving, it is from my side. You can make use of the textbook and you can form your own sentence also. Next, toxins of fungal origin are called mycotoxins. Mycotoxins, just now we have seen fungi, which when obtain the nutrition from the food items, what they do? They release the poisonous chemicals. So mycotoxins are nothing but the poisonous chemicals released into the food by fungi, making the food poisonous and harmful for consumption. I will just wait, so those who want to jot down, they can jot down the points immediately. The first answer is budding. The yeast cell develops small round bodies on the parent cell, which are called as buds, and new daughter cells develop from this buds. Toxins of fungal origin, they are called mycotoxins, fungal origin, which are caused by fungi. So mycotoxins are the poisonous chemicals released into the food by fungi, making the food poisonous and harmful for consumption. And the last is leguminous plants can produce more proteins due to rhizobium. Explanation you can give in your own words. For the process of nitrogen fixation, rhizobia use leguminous plants like beans, sweet pea, soya bean, etc. as the host. Beans and pulses are rich in proteins due to the nitrogenous compounds made available by rhizobia. explanation you will get in the textbook on page number 89, last paragraph of the page. You can just modify the statement and you can write. You can see page number 89, last paragraph they have given in the textbook. Rhizobia produce nitrogenous compounds from atmospheric nitrogen. No need of writing that part. For the process of nitrogen fixation from there up till made available by rhizobia is your explanation. With this, we complete question number 1 and we wind up with our today's session. In our next session tomorrow, I will be discussing the textual exercise and the additional questions for this chapter. If you just go through the questions for this chapter, majority of the questions there is no direct answer in the textbook. You have to make use of the different social media, Google, internet to get the answers. I have made use of the internet and searched the answers for you. It is not compulsory that whatever I have searched that only you have to write. You can write the answers on your own. Only the logic and the application of science is a must. Till then, thank you dear children. Keep completing your notebooks. Do not pile up the work. And your first online assessment is coming near. So be in progress with its preparation. Thank you.